In the realm of wildlife conservation, scientists are constantly seeking innovative methods to track and protect endangered species. One of the most surprising developments in recent years has been the use of leeches as valuable allies in this endeavor. These blood-sucking parasites, often regarded with distaste, have emerged as unexpected heroes in the field of conservation biology. This article explores how leeches are contributing to wildlife research and conservation efforts, and why their role is becoming increasingly important in understanding and protecting biodiversity. The challenge of tracking endangered wildlife. Protecting endangered animals requires a comprehensive understanding of their distribution, habitat preferences, and population dynamics. Traditional methods of wildlife tracking include setting up traps, installing camera traps, collecting hair and fecal samples. While these methods have proven effective, they can be time-consuming, labor-intensive, and sometimes ineffective, especially when dealing with rare species in hard-to-reach habitats like dense jungles. This is where leeches come into play, offering a unique solution to this long-standing challenge. Enter environmental DNA, EDNA. Before delving into the specific role of leeches, it's important to understand the concept of environmental DNA, EDNA. EDNA refers to genetic material left behind by animals in their environment, which can be collected from sources such as soil or water. This genetic information can be analyzed using a technique called DNA barcoding, which involves identifying short DNA sequences unique to specific species. The use of EDNA has revolutionized wildlife surveying, allowing researchers to detect the presence of species without directly observing or capturing them. However, the application of EDNA has expanded beyond soil and water samples, leading to the innovative use of leeches as living DNA collectors. Leeches as living DNA libraries. Leeches, particularly terrestrial species found in tropical forests, have several characteristics that make them ideal for collecting wildlife DNA. Indiscriminate feeding. Leeches are not picky eaters and will feed on a wide variety of animal species. Slow digestion. The blood consumed by leeches can remain in their digestive system for months, preserving DNA from multiple species over time. Easy collection. Researchers can easily collect leeches by simply walking through the forest, as the leeches naturally attach themselves to passing animals or researchers. These properties make leeches natural repositories of genetic information from various species in their habitat, earning them the moniker of living DNA libraries. Pioneering research. The 2012 Vietnam study. The groundbreaking potential of using leeches for wildlife surveys was first demonstrated in a 2012 study conducted in the tropical forests of Vietnam. Researchers collected 25 terrestrial leeches and analyzed the DNA found in their stomach contents. The results were remarkable. DNA from 6 different mammal species was identified. Some of these species had not been previously recorded in the area. The study detected the presence of Annamite-striped rabbits, which had eluded detection even after more than 2,000 nights of camera trap surveillance. This study introduced the concept of invertebrate-derived DNA, iDNA, and showcased the potential of leeches as effective tools for wildlife surveying, especially for elusive species. Expanding the technique. Subsequent studies. Following the success of the Vietnam study, researchers have continued to explore and refine the use of leeches in wildlife conservation. Southeast Asia Study 2018. Over 700 leeches were collected across Southeast Asia. DNA from dozens of bird and mammal species was identified. The study revealed that leeches are particularly effective at detecting small mammals like rodents and shrews, which are often difficult to survey using traditional methods. Surprisingly, bat DNA was found in one leech, expanding our understanding of leech feeding habits. China Nature Reserve Study 2022. DNA from over 30,000 leeches was analyzed. Park rangers assisted in collecting leeches during their regular patrols. The study successfully identified DNA from threatened and endangered species. Researchers were able to map the spatial distribution of species across the park, revealing that native species were more common in the park's interior and less frequent in areas with high human activity. Borneo Study. Similar to the China study, this research found that leech-derived DNA showed lower wildlife diversity in areas disturbed by human activities such as logging. These studies demonstrate the power of leech-derived DNA in not only detecting the presence of rare species, but also in understanding how human activities impact wildlife distribution and diversity. Advantages of leech-derived DNA. The use of leeches in wildlife surveying offers several advantages over traditional methods. Ease of collection. Researchers can passively collect leeches while conducting other fieldwork. Access to elusive species. Leeches can provide DNA from species that are difficult to detect using other survey methods. Non-invasive sampling. Unlike trapping or direct observation, collecting leeches doesn't require disturbing the animals being studied. Long-term DNA preservation. The slow digestion of leeches allows for the detection of species that may have passed through an area months before the leech was collected. Cost-effective. Leech collection requires minimal equipment and can be integrated into existing conservation activities. Limitations and considerations. While leech-derived DNA has shown great promise, it's important to acknowledge its limitations. Feeding preferences. Not all animals in an ecosystem may be equally represented in leech samples due to differences in leech feeding habits. Spatial accuracy. The movement of leeches after feeding can make it challenging to pinpoint exact locations of detected species. Protected leech species. Some medicinal leeches are themselves protected, limiting their use in these studies. Incomplete picture. Leech-derived DNA should be used in conjunction with other survey methods for a comprehensive understanding of biodiversity. Beyond leeches. Other invertebrate DNA sources. The success of leech-derived DNA has inspired researchers to explore other invertebrates as potential sources of wildlife DNA. Flies, beetles, ticks, mosquitoes. These insects, which feed on blood, dung, or carrion, can also serve as natural collectors of genetic material from various species. Future directions and potential applications. The use of leech-derived DNA in wildlife conservation is still a relatively new field with exciting potential for future applications. Disease tracking. Leeches might contain DNA from pathogens in the blood of their prey, potentially helping track the spread of wildlife diseases. Refinement of sampling techniques. Improving our understanding of leech behavior and feeding patterns can enhance the accuracy and reliability of iDNA surveys. Integration with other technologies. Combining leech-derived DNA with other survey methods and advanced data analysis techniques could provide even more comprehensive insights into ecosystem health and biodiversity. Expansion to new habitats. While most studies have focused on tropical forests, the technique could be adapted for use in other ecosystems where blood-feeding invertebrates are present. Conclusion. The use of leeches in wildlife conservation represents a fascinating convergence of parasitology and conservation biology. By turning these often maligned creatures into allies, researchers have gained a powerful new tool in their efforts to understand and protect endangered species. As we face unprecedented challenges in biodiversity conservation, innovative approaches like leech-derived DNA offer hope for more effective and efficient wildlife monitoring. While not a complete solution on its own, this technique adds another valuable method to the conservationist toolkit. The next time you encounter a leech in a forest, consider the wealth of genetic information it might contain. A tiny wriggling time capsule of the forest's biodiversity. In the ongoing struggle to protect our planet's endangered species, these unlikely heroes are proving that sometimes, the most valuable allies can come from the most unexpected places.